Comparative Placentation by Group 8. Before the discussion proper, there are two questions we need to ponder. First is, how does the embryo manage to live inside the womb? Second, what is a placenta? So upon entering the uterine cavity secretes uterine milk, which serves as the initial nourishment for the embryo. But because the embryo is rapidly developing, you will find that the histotrough quickly becomes insufficient for the nourishment of the embryo. What should happen is that there should be a close relationship between the extraembryonic tissues and the maternal circulatory system. These action would allow the embryo to receive blood-borne maternal nutrients, also called hemostroph, from its mother. Meaning to say that inside the uterus, the embryo does not physically eat or drink, but instead it gets nutrients from the blood of its mother. Together, histotroph and hemotroph are what we call embryotroph. Now that we know this, how do these nutrients reach the embryo or the fetus? So these nutrients reach the embryo or the fetus by a temporary organ that connects the maternal tissues and the extraembryonic tissues, and this is called the placenta. The job of the placenta is to necessitate close synchrony between the state of uterine receptivity and the stage of embryonic development. Placenta is formed through the process of placentation. As we recall, in the prior reports, there is the mention of the zona pellucida shedding. And the moment that the zona pellucida is shed, the endometrial epithelium of the uterus is penetrated by the embryo and where it becomes completely embedded. And this is called implantation. To the right is a picture that depicts the development of the zygote until it reaches the blastocyst implanting and the precise location of where they are during that time. So the cleavage becomes the morula, then it becomes blastocyst, and then it implants itself. There are two kinds of implantation. First is the invasive and second is the non-invasive. In invasive implantation, the trophectoderm multiplies and embeds itself deeper into the endometrial epithelium. And soon enough, the endometrium cells around the embryo divide and get larger until the endometrium completely surrounds the embryo. This is found in rats, primates, and carnivores. In non-invasive implantation, however, the trophectoderm does not embed itself deeper and only attaches itself to the uterine lining. This is found in domestic animals except carnivores. This picture describes the process of invasive and non-invasive implantation. First, before it starts, there is the blastulation. As you can see in A, this represents the fertilized zygote or the fertilized egg rather, and in B and C, this represents the zygote undergoing cleavage, and in D and E undergoing blastulation, and in F, this is the blastulated cell. After that, the blastulated cell can undergo either invasive implantation or non-invasive implantation. In invasive implantation, first the blastulated cell comes in contact with the endometrial epithelium cell or the lining of the uterus. And then the trophectoderm multiplies and embeds itself further into the uterine lining or the uterine wall. And then it gradually invades or implants itself inside the uterine wall, wherein the next phase would be it would be wrapped around, and this is where the embryo would be located for the rest of the gestation or the pregnancy. In non-invasive implantation, however, the trophectoderm does not embed itself deeper like in this dissolution. Instead, it only attaches to the endometrial lining. And to prevent this attachment, there is the interdigitated microvilli which helps the blastulated cell or the embryo or fetus to remain attached to the lining of the uterus throughout gestation. Next is the peri-implantation conceptus development. The embryo reaches the uterus prior to blastulation, and by the end of the blastulation, the embryo would consist of the trophectoderm, inner cell mass, and the blastocell. A thing to note is that when trophectoderm is involved in placentation, it will be called trophoblast. The trophoblast is expelled at parturition as a part of the afterbirth. So let's go to what happens to the inner cell mass. Around the time that the embryo hatches from the zona pellucida, the ICM differentiates into the epiblast and the hypoblast. The hypoblast gradually forms the lining of the primitive yolk sac. Meanwhile, the epiblast undergoes gastrulation to establish the three somatic germ layers, which are endoderm, mesoderm, and ectoderm. The three germ layers. What happens to the three germ layers during placentation? Endoderm gradually displaces the hypoblast beneath the epiblast, and the extraembryonic mesoderm is divided into the somatic and visceral sheaths. Somatic or parietal sheath associates with the trophectoderm to form the chorion. Meanwhile, the visceral or splanchnic lines the extraembryonic column. The visceral or splanchnic mesoderm also associates with the hypoblast and the endoderm to form the splanchnoplura. At the same time, the primitive yolk sac, which was formed from the hypoblast, is developed into the primitive gut and the definitive yolk sac. The primitive gut is a flat sheet of intraembryonic endoderm that will change into a tubular gut due to the folding of the embryonic body, head, tail, and lateral body folds. The definitive yolk sac, in some species, fuses with the chorion to form the choriovitelline placenta, which will be discussed later. The allantois develops an invagination from the hindgut after the formation of the definitive yolk sac. Hindgut has an inner wall of endoderm and an outer covering of visceral mesoderm. The visceral mesoderm of the yolk sac is where blood and blood vessel formation is first seen. This is followed by the vascularization associated with the allantois. The somatic mesoderm, however, remains avascular initially. As the embryo grows, the allantois expands into the extraembryonic coelom, eventually occupying majority of the cavity. Where the allantoic wall and the chorion meet, they form the chorioallantois, which vascularized from the visceral mesoderm and gives rise to the chorioallantoic placenta, which will also be discussed later. So this is the development of the extraembryonic membranes. As you can see, infant starts with blastulation, and then it undergoes the differentiation of the inner cell mass into the epiblast, which is number 4, and the hypoblast, which is number 5. Also, the hypoblast, um, it it divides further until the two ends meet, which will then form number 10, which is the primitive yolk sac together with the endoderm. In C, you can also see the zygote after gastrulation, in where you can see that it has gastrulated and there is the three somatic germ layers. In D, as you can see, the extraembryonic coelom is starting to surround the definitive yolk sac, and also the definitive yolk sac has formed from the primitive gut, and as you can see, the allantois is also forming. And in figure E, the allantois has formed, and also the splanchnoplura, which is formed by the allantois and the extraembryonic coelom. And also, this is the fusion of the yolk sac wall and the chorion, which will then lead to cardiovitaline placenta. And the next is the allantois. It expands until it has covered most of the extraembryonic coelom. And in G, as you can see, the amniotic cavity has already formed, and there is also the mesamnion. And as you can see, it is surrounded by the allantois. And in this part where the allantois comes in contact with the chorion, is what leads to the formation of the chorioallantoic placenta. In cattle and pigs, the allantois does not completely surround the amnion. Rather, there is a part where it is not found, this part. However, in horses and dogs, the allantois surrounds the amnion completely. Changes in the endometrium and maternal recognition of pregnancy. As the embryo enters the uterine cavity, the uterus prepares for placentation by producing progesterone when the early embryo moves from the oviduct to the uterus. This stimulates marked changes in the endometrium, which is the lining of the uterus. So, to understand further, let us see what happens to the uterus during the estrus cycle. During the proestrus to estrus, high levels of estrogen are produced. This induces the proliferation of the uterine glands and the simultaneous engorgement of the stroma with blood and extracellular fluid. This action causes edema. In met-estrus, the endometrial edema decreases and some of the congested blood vessels break down. This causes the proliferation of the uterine glands to continue. And in diestrus, the glands reach maximal secretory activity, which provides histotroph, or what we call the uterine milk. Luteolysis, which is the regression of the corpus luteum, is a process that needs to be prevented in order to continue pregnancy. Luteolysis is based upon the recognition of maternal pregnancy, which means that if the mother fails to recognize that she is pregnant, this could lead to termination of the pregnancy. So, in order to prevent this, the embryo signals the uterus of its presence during implantation. But the effectiveness of the signaling depends on the degree of the contact of the trophectoderm and the endometrium. In ruminants, corpus luteum produces both oxytocin and progesterone. Oxytocin stimulates the uterus to produce prostaglandin F2 alpha, which, as we recall, stimulates the regression of the corpus luteum, which should be avoided to continue pregnancy. However, the trophectoderm, upon contact with the endometrium, forms or produces interferon tau, which inhibits the formation of oxytocin receptors in the endometrium. Interferon tau also stimulates histotroph production. This means that if the trophectoderm releases IFNT, oxytocin cannot stimulate the production of PGF2 alpha, preventing luteolysis, which would continue the pregnancy. In pigs, however, it is completely different because the trophectoderm releases or produces estradiol around days 11 to 12, which causes the production or the release of prostaglandin F2A into the uterine lumen, which is exocrine, instead of the maternal bloodstream, which is endocrine. Estradiol is also believed to stimulate myometrial contraction, which facilitates the distribution of multiple embryos within the pig's long uterine horn. Also, there should be at least 4 embryos to prevent the teolysis. Any less, and there would be the regression of the corpus luteum, which terminates pregnancy. In mares, however, it is very different because direct cell-to-cell contact between the trophoblast and the endometrium is prevented until day 21 by a capsule. This enables the horse embryo, which is, as we recall from the first chapter, it is spherical and it migrates over the entire endometrial surface between 12 to 14 times per day during 6 to 17 days of pregnancy to prevent the teolysis. In mares, it is believed that this is the form of signaling instead of like in other species, which is implantation. The role of signals exchange in mare are still not understood, but what is known is that this migration process is important. In carnivores, the signal of maternal recognition of pregnancy has yet to be identified. Because the canine estrus cycle takes too long, a non-pregnant bitch sometimes develops a syndrome called pseudopregnancy. Pseudopregnancy is when the corpus luteum continues to produce progesterone even if the mother is not pregnant. This causes the bitch or the mother dog to exhibit pregnancy symptoms even if there is no zygote or there is no fertilized egg. This condition mostly reverses by itself, but sometimes it may need treatment. Moving on to the classifications of placenta. There are multiple ways on how to classify a placenta, and this is the first one. Based on the nature of extraembryonic tissues of placenta. First is the choriovitelline placenta, which is formed when the yolk sac wall and the chorion fuses. And this is only functional in carnivores and horses. These are never functional in ruminants because the yolk sac involutes or it regresses 3 to 4 weeks after conception. Second is the chorioallantoic placenta, which is the fusion of the allantoic wall and the chorion, and it is primarily the functional placenta in domestic species. So this is the difference between the two. As you can see in choriovitelline placenta, there is an enlarged yolk sac. This is the yolk sac. And it fuses with the chorion, which is this area. And in chorioallantoic placenta, instead of the yolk being enlarged, as you can see here, it is the allantois that is large. And there is also the presence of villi, which helps to embed deeply into the uterus. Second classification is based on interaction with endometrium. First is the chorion frondosum. In chorion frondosum, chorioallantois interacts with endometrium and is engaged in placentation. It has villi, therefore it is not smooth. It does not have a smooth surface. Second is the chorion levy, which is very different from chorion frondosum because chorioallantois is free. It does not interact with the endometrium and it is not engaged in placentation. Because of this, it has a smooth surface. This part is the chorion frondosum. As you can see, these are the villi. And this is the chorion levy. There are no villi, hence why we said that it has a smooth surface. Next classification is based on the endometrium. First, in pigs and horses, the chorion frondosum is diffusely distributed over the entire chorioallantoic surface. This is called diffuse placenta. In porcine, chorioallantoic surface area is increased through foldings. There are two kinds of foldings, which is the primary or plique or the secondary or rugae. Third is the cotyledonary, which can also be called multiplex and villus, and it is found in ruminants wherein the chorion frondosum is organized as arborizing chorionic villi assembled into larger macroscopically visible tufts called cotyledons. Chorion levee is found in between cotyledons. These cotyledons combine with the endometrial prominences, also called caruncles, forming placentomes in which the chorioallantoic villi of the cotyledons extend into the crypts of the caruncle. Fourth is the zonary and lamellar. In carnivores, chorion frondosum is organized into a broad belt extending around the longitudinal axis of the embryo where it forms the lamellae. Next, classification is based on the number of tissue layers separating the fetal and maternal circulations. This is also called the placental barrier. There are three extraembryonic layers that are always in chorioallantoic placenta. First is the endothelium, which is the lining of the allantoic blood vessels. Second is the chorioallantoic mesenchyme, which originates from the fusion of somatic or chorionic mesoderm and the visceral or allantoic mesoderm. And third is the chronic epithelium. The number of layers, however, that is retained depends on the species. Before placentations, the endometrium presents three layers that contribute to the placental barrier. First is the endometrial epithelium, second, connective tissue, and third is the vascular endothelium. In domestic animals, there are two main placental classes according to the number of layers in the placental barrier. First is the epitheliochorial placenta, which can be seen in pigs, horses, and ruminants. In here, chorionic and endometrial tissues are opposed, but there is no loss of maternal tissue. There can also be synaptiatichorial placenta, found in ruminants, where endometrium is modified and crossed into by trophoblast cells. The second type is the endotheliocorial placenta, which is found in carnivores. In here, the endometrial epithelium and connective tissue are lost during placentation, leaving the trophoblast in direct contact with the maternal vascular endothelium. A type of this is the hemocorial placenta. This is found in rodents and humans, wherein there is a reduction of the placental barrier and the trophoblast is in direct contact with the blood. Also included here is the deciduate placenta and adeciduate placenta. In carnivores, which is deciduate placenta, there is a tight adherence between the fetal and maternal components of the placenta. This is why a portion of the portion of the endometrium is called decidua, is also shed upon parturition as part of the afterbirth. In a deciduate placenta, however, there is no decidua, meaning to say that there is no part of the endometrium that is shed upon parturition as a part of the afterbirth.